be recording. All right, so today what we're going to do is talk about ketone bodies, and we may get a little bit into amino acid metabolism. Ketone bodies are they're energy-containing molecules, we could say, or they're energy molecules that are produced by the liver when the body's low on carbohydrates. I'm going to just call them energy molecules, I guess. Okay, so conditions where this occurs during starvation. All right, so you can't get enough resources. It occurs if you're on a low carb diet. Okay, you might be taking in lots of fats and proteins and no carbs, and you're not going to be getting those. The energy, it's still the same sort of thing. You're not going to be able to put glucose in your blood. Your brain runs on glucose. Your heart likes to run on glucose at sometimes, or partially on glucose, but really, a large part it's your brain. You go on a low carb diet. I've never done it, but what I've heard is, it's hard to you're a little bit fuzzy headed at the beginning of it until your body adjusts. That's what I've heard from people on that, and it also happens during diabetes. Okay, there may be other cases of where it happens, but these are some good examples for us. What's happening during diabetes is you might have lots of carbohydrates in your body, but because you are insulin resistant, if it's type 2 diabetes, you're insulin resistant, you can't see the insulin even though it's there. If you're type 1 diabetes, you're not making insulin, okay, and it's not there. So the liver thinks that you have no carbs and you're starving, and it needs to make ketone bodies. All right. We've talked about the mechanism that you can use to make, so we can take carbs and make them into fats, right? Because fats are made with acetyl-CoA, and if you break down glucose, you get acetyl-CoA, right? So you can make fats from carbs. There is no pathway back the other way. Okay, so you can't make glucose from fats, or from most of the energy in fats. So what we have here is the pathway to make ketone bodies. I want you to take a look at it, see the whole thing there. And then what the ketone bodies are, essentially they're being made from acetyl-CoA. And where can we get acetyl-CoA when we don't have carbs? Beta oxidation. Beta oxidation. We get them from fats, right? Fats. I should say triacylglycerols. So let's go ahead and look at this process in detail. And we're going to do like our ATP counting thing as usual. How much energy is used in this process?
Do you want us to know the steps?
Nope. No. You just need it. What you need to do, what you really need to know is you need to, recognize the pathway. Recognize the pathway. Discuss purpose. Okay. Recognize the products of the pathway, which are ketone bodies. Okay. The ketone bodies. There's two of them. You don't even need to give their specific names. You just have to say those are ketone bodies and this is what they do. This is what they're for. Okay, so that's where we are with this pathway. I like this one because as we're going to see, well, I like this one because there's something interesting that happens with energy with this one. We'll see. We'll go through. All right, so we've got two acetyl-CoA. What the liver does is it pulls them together. It makes an acetyl-CoA. It comes in. It takes another acetyl-CoA in. So far we've invested 3 acetyl-CoA, right? And then what happens? We get this acetyl-CoA comes off. So now we're back to 2-acetyl-CoA, right? We put it in acetyl-CoA, we got it back, and then we get acetoacetate. This is one of our ketone bodies, all right? And it could also be further, what's going on? Is it being reduced or oxidized here? To make beta hydroxybutyrate. It's being reduced, right? So we can further reduce it. So this, you can get more energy out of this than you can get out of this, right? Either of these can go to your blood, where they're then going to travel to the heart and the brain. The brain's really at the worst in the worst case here, right? Worst at odds here, because the brain does not run on fats at all. Okay, no fatty acids in the brain, and so to the blood. Okay. What's also a problem with acetoacetate here is in the blood, you can actually have it break down to make acetone. Okay, so you can lose this carbon dioxide here. And this is why if you have somebody who has untreated diabetes, their breath will smell sweet. You may have heard of that as a symptom, and it's because they're actually making a bunch of ketone bodies, and they're ending up with some acetone in their blood. And you're smelling the acetone. Yes.
I got behind. Where is the third acetyl-CoA coming from?
So the third acetyl-CoA. Well, where is it coming from?
Like, where is it in the mechanism? I only saw it too.
So let's go ahead. We got this much. Sorry, I have to blow it up to make it so we can see it. So here's our third acetyl-CoA comes in.
I see it now.
Okay.
Yep.
But we get it right back. Right?
Mm-hmm.
So we build things up. The mechanism is such to make our acetoacetate, we need to make this, and then we'll get it back in terms of acetyl-CoA. So we get that acetyl-CoA out. So 2-acetyl-CoA went in, net, right, to make this. And then in terms of NADH, we've got one here. Let's scoot back up. Were there any more? Okay, that's it, right? So if we're making this is 2-acetyl-CoA and this is 2-acetyl-CoA and an NADH. All right, so you need to know these two structures. Yes? Those are the ketone bodies. If I show you a structure, one of them, you need to tell me it's a ketone body. You don't have to give me you don't have to give me the specific name. And you have to be able to tell me what ketone bodies are for, which is what? Why do they get made?
You have no carbohydrates.
You don't have carbohydrates. They're made as an energy source for the tissues, right? Your liver is responsible for dumping stuff into blood so your heart and your brain and all those things get enough energy. Okay. If you take farm chem, your liver is responsible for getting rid of xenobiotics, right? Metabolizing drugs. But your liver does a lot of stuff. It's an important organ. But here we care mostly about the energy. All right, so this gets to your brain, let's say. Let's use your brain as the example. So from the blood, your brain gets either of these. They both can go in the blood. Okay, so let's say they come in your brain. We get the hydroxybutyrate comes in, let's say. So that was our most produced species. What happens with the NADH? You get it right back. Okay, but it was spent in the liver and you're getting it back in the brain. All right, so by getting this, your brain gets an NADH. I expect your brain is happy about that. But it's also happy with the acetoacetate, given there are no carbohydrates around. Okay, and then we're going to go ahead and travel down this pathway. We got our NADH back if it was spent, and then in the end we get how many acetyl-CoA out? 2. How many did the liver put in? 2. So how many, if we just think about the two pathways here, we ran the pathway, right? We built them up, and we tore, and that cost us 2 acetyl-CoA and 1 NADH, right? And then we broke them down and we got back 1 NADH and 2 acetyl-CoA. So what's the difference in breaking them down versus building them up? 0. We don't see that very often. Usually it costs more to build something than you get back breaking it down. Okay? So that's an interesting thing about this pathway. Now you do spend the energy pumping this stuff around in your blood, right? But yes.
So what prevents it from just happening at the same time? Like in the other ones, it was prevented because it was a futile cycle. So what stops it here?
Right. So what happens? What prevents you from having a futile cycle? I don't actually know for sure, but there's no reason. There is no reason for the liver to break down ketone bodies, right? Because the liver, if it's got the fats, it's just going to use them if it needs it for energy. So they're energy molecules. There's no reason for the liver to break them down. So the liver may not even have this the reverse pathway, and the brain is never going to be making ketone bodies, nor your muscles. They don't do that. So I'm guessing. My guess is that it's organ compartmentalization. Yeah, Daniel.
Couldn't you, so you start with acetyl-CoA, couldn't you just run that through citric acid cycle and get oxaloacetate and then make glucose? Why would it do this instead?
So okay, that's a great question. Let's go find ourselves in a citric acid cycle picture.
So you get acetyl-CoA, and then why wouldn't you just run it back up through gluconeogenesis after it's converted to oxaloacetate?
Okay, so first off, before we go to the citric acid cycle, we don't have a pathway to take our acetyl-CoA, right, and make it back to pyruvate. So we can't make oxaloacetate that way,
Okay.
right?
You get oxaloacetate from the citric acid cycle. Right?
Yep. I need a diagram. It's a great question. It's electron transport chain, isn't it? Yeah. Where's our citric acid cycle? I'm pretty sure we talked about it.
We did. It would probably be after that.
That's lab.
Because
It's
too far back because we talked about it.
But we wouldn't have talked about. I had hypothyroidism. We wouldn't have talked about. Here it is. No, it isn't. It's got to be right after this.
I think that was lab.
This is lab. Okay.
Yeah.
How about the end of this? Haha, there it is. All right. Citric acid cycle. We put an acetyl-CoA in, right? How many carbons? 2. I didn't tell the answer, sorry. How many carbons? How many carbon dioxide come out?
2.
2. So you can't make more oxaloacetate because every 2 carbons you put in, you get 2 out. We can't build up our oxaloacetate in this perspective. The pathways just don't exist, and that's why you have to make ketone bodies. You can't take fats and make them into carbohydrates. Evolution, and the answer, why is that? Well, natural selection. I don't have a better answer, but yes.
So why aren't ketone bodies bad for you?
So if you're making ketone bodies, right, it's signaling a bad state, right? Starvation is bad. Low carb diet may or may not be bad. And then, diabetes is bad, right? So both of those, both natural things there are bad. There are problems people talk about. I've seen cases where somebody goes on this, they just switch off of carbs exactly. I've seen reports of, and they immediately get this ketosis. What's the exact name, Bridget, you mentioned?
Diabetic ketoacidosis.
Okay, so ketoacidosis, not the diabetic version, that somebody just switches too fast and their body can't handle it. Yeah.
What about the production of this is like not, or like not as good for you? Do you mean like it's just a different pathway? Why is it?
Right. Why is it bad for you? I'm going to ask Bridget if she knows why it's bad for you. Your blood becomes too acidic, would kind of sound like it, like your pH in your blood would get too low, but I don't know why exactly it's bad for you, and I don't know that it is possibly all that bad for you. Is it okay?
It can cause, I don't know exactly the mechanisms, but it is a pH issue. That's why the first thing they do if they suspect diabetic ketoacidosis is take a arterial blood gas and check the pH. But it can cause your body to shut down and go into a coma.
But why?
Yeah, and I've heard
Because
that.
the body's
I don't.
trying to preserve like itself. And so it's like, okay, I'm gonna just shut off every non-essential function other than keeping my brain alive.
I'm gonna. I'll look that. I'll look it up and try and get something back to folks on that. Okay. See if we can get something more specific.
Yeah, that's
But it's also a natural process, right? This is what you do when you don't have enough carbohydrates around. So it's bad in some cases. I don't know that it is supremely bad. If that makes sense.
the dose makes the poison essentially.
Okay. So questions on that process, though. So what you know, once again, recognize those ketone bodies, know what they're for, okay, and know when they occur. All right, I've got this picture that I had my notes from before, so I just wanted to show you an adipocyte because I think they're cute. But I mean, if you look at this, the emphasis here on adipocytes is they're just bags of fat, essentially. So, I mean, this one doesn't even look as exaggerated as other ones I've seen. You've got the nucleus of the cell. You've got the cytoplasm. It's like this little layer around it. And this is all fat. It's just fat storage. So when I say fat, I mean triacylglycerols. So they get made for storage. Your lipids are going to be made into triacylglycerols, which are super nonpolar, and they all go in here and stick together, and they're in this bag, which is the adipocyte. Okay. Now, when they need to be mobilized, this is an example of that process. And there's some interesting things to talk about here. All right, so what we've got up at the top here is this is a G-coupled protein receptor. And so we have some hormone that binds here. Let's say it's glucagon. Why would glucagon cause you to release fats?
Low blood sugar.
Low blood sugar, right, okay. Diabetes, the same thing's gonna sort of happen, right? Because you've got insulin in your blood, it doesn't recognize it, you actually do mobilize fats for these sorts of things, fat reserves. So you have this hormone, it's gonna bind there, and that's gonna cause cyclic AMP to be made, okay? So I'll go ahead and I'll write out the steps over here. A hormone, glucagon, is an example. Adrenaline's another example. Why would adrenaline cause you to release fats?
You need energy to run away from something.
Yes, you need energy to run away from something or fight something, right? Binds to the receptor and activates the formation of cyclic AMP. Okay, and this is what we call a second messenger. We haven't done the signaling chapter yet, but I'll just say it now and then we'll talk about it again. Repeating things is always good for helping people with it. But a second messenger is something where we've got the primary messenger is whatever hormone binds on the outside of the cell, and then something gets made in the cell that's going to cause changes. And what we see here for this cyclic AMP being made is that it's going to cause fats to be released, right? But it's also going to cause glycogen to be broken down. We'll have those sorts of things going on, right? The cyclic AMP is going to signal a bunch of different things. Okay, it's going to turn on a bunch of different pathways because you don't just release fats when glucagon's in the blood. All right, so we have that cyclic AMP is made, all right, which is going to activate protein kinase A, which, so first off, protein, it's called protein kinase A. That's this pKa. Let me go ahead and abbreviate it here as pKa as well. How do you think it activates protein kinase A, cyclic AMP? Okay, phosphorylates. It's a decent guess. It's not correct.
Isn't it's it's a cyclic AMP activated kinase that does the switching from the PF2 activate inactive and
You tell me I guess there are multiple steps there.
it's
Yeah, so multiple steps here. So what I was thinking is actually incorrect. Phosphorylation is also incorrect. But so we've got multiple. Since we got a dashed arrow, we got multiple steps in some sort of cascade. But if it was just one arrow, it's going to bind to it and it's going to cause it to change shape, right? So if this directly interacted, that would be the case. But Bridget's right that there's going to, since we have a dashed arrow there, we have more things, which activates. Okay, and so we have the protein kinase A. The second question is protein kinase A. What does it do?
Phosphorylates.
It phosphorylates something, right? Because it's a kinase. Okay, which phosphorylates this perilipin. So perilipin is a protein here that's on the edge, and we're just going to look at the cartoon here, right? Perilipin is coating the outside of this lipid droplet, and where the lipid droplet is, when we go back and look at our lipid, right? The lipid droplet is this, right? Okay. So what on the outside of that, you have this protein that's called perilipin. All right. If we were to speculate, we don't have the structure, and I'm not going to bother with the structure, but if I look at this and I was to speculate, what would this side of the perilipin look like? What properties would it have? It would be nonpolar. We have nonpolar amino acids here, right? Because it's facing the lipid. What types of amino acids do we have on this side, the upper side? We'd have polar, right? Polar charge, whatever we need for there. So what happens is this gets phosphorylated and that causes a cartoon of it, okay? But that's going to cause it to stand up and give us some space so that we can actually get some fatty acids out, okay? I'm going to say which changes shape, which changes shape and allows lipids to leave the droplet. Let's say triacylglycerols, because that's what they are. Okay, those triacylglycerols are then broken down to make fatty acids. Okay. Which can then enter the blood. I may have gotten my numbers off here. Let's take a look at this. Actually, I want to revise what I said. We're leaving room for change shape allows the triglycerols to leave the droplet, but really what it is it allows this hormone-sensitive lipase to come in and join the droplet, I guess. Allows the hormone-sensitive lipase And it's going to convert those triacylglycerols into fatty acids that leave the droplet. That looks like that's all of 5.
3 doesn't seem to match.
Doesn't it? We'll go up and fix it.
Okay.
Right? Those fatty acids, while they're not super soluble, are much more soluble in water than the triacylglycerols. Let's go back and look at 3. Let me get my glasses. Okay. So 3 says, which activates the protein kinase? Because there's a 4 there. Oh, 3 is for the hormone sensitive lipase.
Yeah.
Shoot. Well, here, we're going to make this 4, which is going to mess up everything else, isn't it?
Mm-hmm.
How about I say really 4? And then this is going to be, we're phosphorylating periolipin as part of 4 as well, right?
Mm-hmm.
And 3 is about activating the, and this is going to be and 3, okay, in terms of that, because 3 is active, we're going to, the protein kinase A is phosphorylating hormone sensitive lipase as well, and it's going to cause that we have some movement here when these things stand up or change shape, really. That's a cartoon for it. And then we get these fatty acids that get out. And then for 6, they're going to fatty acids which enter the blood.
So the 1, 2.
Fatty acids enter the blood, right? Sophia, do you want to make a comment about?
Well, okay, with 1, 2. Can you just like say the order of them? Like 1, 2, 5, 3, 4.
Like change these numbers here? I think it's going to blow up if I do that,
well,
but
it's 5.
because it's actually.
Like the steps on, like the numbers correlated with what you wrote to what's on there, like put a different number next to that.
Like ABCD.
It might
No,
make,
shall I just do it and I'll write it? How's that?
make more sense to have which
1.
activates protein kinase A as like a 2.5, and then have 3 phosphorylates hormone sensitive lipase, 4 phosphorylates for perlipin.
Okay.
Is that okay? So in terms of this, we're talking about the process. I would like you to be able to look at a figure like this and interpret it. Does that make sense for this? So this is not going to be, hey, I want you to reproduce this figure. Okay, yes.
This is for the breakdown of the adipocytes.
This is breakdown of fat. So you've got fat stored in a adipocyte and you have to get them out, right? And they have to go someplace. Okay, Daniel.
You said free fatty acids are more soluble in water. Wouldn't the triacylglycerol be because it has more hydrogen bond donors, etc.?
It doesn't have any donors. It only has acceptors with the esters, and they're not even all that great.
What about the OHs? Oh wait, never mind.
Okay,
I'm good.
it's okay.
I had to think about it.
But the fatty acids have a charged end, so they're better. I can tell you from having worked with them in labs, some they're still hard to get into solution. And they're hard to get into solution. That's why you need a carrier protein in the blood. So fatty acids enter the blood and stick to a carrier protein. We'll just say stick to serum albumin. Okay, I'm going to come back to that one. And then fatty acids are going to then enter cells and be broken down. Okay. Or specific transporters. It's oxidized to make ATP. Okay, the cell they're talking about here is a myocyte. It would be anything that could use fats. So an interesting thing with the serum albumin is this is one of those places that you end up with drug interactions. So serum albumin is this soluble protein, but it also has hydrophobic patches on it where your fatty acids can stick, okay? But not only can fatty acids stick to serum albumin, but if you're taking any number of pharmaceuticals that are kind of on the borderline between being really water soluble, right? Remember, you're designing one of these, you want them to go through the cell membrane easily, so you're going to try and make your drugs as hydrophobic as possible, but they still have to go in the blood, which they have to be hydrophilic to get into the blood. So you have to kind of try and straddle that line, try and come as close as you can to the line between hydrophobic and hydrophilic. And what that means is there are a number of pharmaceuticals that can load up onto serum albumin. Okay? And so the actual, if you think about the accessible concentration of whatever that drug is in your blood, that accessible concentration is the amount of that drug that is not on serum albumin. Does that make sense? So we could think about a drug, and so we've got serum albumin plus drug, and we've got an equilibrium with where then we have serum albumin drug. Okay, and you might have it be 95% of that of your drug, and 5% of your drug is out here in solution. Okay, so then the effective concentration of that drug for what you're being treated is 5%. So you're taking it, you're fine. And then you take another drug that also binds to serum albumin. And what it does is it occupies that site, and so it knocks that off, this drug off, and now maybe this drug is 10 or 15% is in solution. You've doubled or tripled your dose. So that's one thing they need to worry about with drug-drug interactions is how much is this thing on serum albumin. Okay, does that make sense? We're about to move on we're going to start amino acid metabolism. Any questions before we go?
What chapter is that?
No idea.
Okay. Yeah.
All right. So what I want to start off then, so we're going to be talking about amino acid metabolism. And if I wanted to talk about it, I would say is complex. All right, you've got 20 different amino acids, and you want to break them down. You want it to be convergent, right? So you're going to try to, when amino acids are broken down, you're going to try, the body tries as much as possible make them into acetyl-CoA or pyruvate. Okay, which is an example of what characteristic of catabolism? It's convergent, right? We're trying to make these into all the same thing. But there's a problem with that amino acids have what on them that isn't in pyruvate? Nitrogen, right? Okay, so for this to occur, nitrogen must be removed. All right, so we're going to talk about, a good part of this chapter is going to be talking about how we deal with that nitrogen. But before we talk about that, what I want to do is talk about nitrogen fixation, or how the process today of how organisms will capture that any free nitrogen. Okay, so nitrogen, I should say fixed nitrogen, because the nitrogen in the atmosphere is not toxic to us, or it's really unreactive. Fixed nitrogen is either very scarce and thus valuable, or too abundant and toxic. All right, so you don't want free fixed nitrogen just floating around. You don't want, if you're any particular number of different organisms, if you're a plant and you've got free nitrogen around, you want to capture that, right? In most cases, because as a plant, you can't walk and go find some other nitrogen elsewhere. All right, for us, especially if you're, especially in the developed world, nitrogen actually is toxic if you get too much nitrogen. You get enough protein, okay? And so you have excess nitrogen, you have to have ways to capture it and not have it become toxic. And so when we think about it, we can think about unfixed nitrogen. All right, unfixed nitrogen is just N2, right? I think it's 79% of the atmosphere. All right, so lots and lots of unfixed nitrogen. That is not a problem. Fixed nitrogen, we talk about fixed nitrogen. This is nitrogen that's biologically available to all organisms. And so examples of that are ammonia, which is pretty darn toxic. That's NO3 minus, okay? That's NO2 with nitrates, okay? That's amino acids, all right? And etc. All right, and other things, all right? So we've got these various forms of fixed nitrogen. And essentially, we're going to talk about what happens with this. All right, so if I take an amino acid and I just directly knock off that nitrogen, it's going to come off as ammonia. And you can incorporate ammonia in the environment. And so if you're an organism like a plant that can't move around or a bacterium that needs to get nitrogen from somewhere, right? Or your yeast in one of the things I'm brewing, you know, you want to pick up nitrogen. I actually add diamino phosphate. So I'm adding NH4, I don't put the charges on there. I add this when I'm brewing. What, this is usually -3, so we'll have to put an H. Okay. So that my little yeast all have enough ammonia and they have enough phosphate so they don't get angry and make bad things. And so what those yeast are going to do is they're going to take that ammonia, they want to capture it, and they're going to combine it with glutamate.
Just yeast,
No, everything.
everything.
Everything, you either are doing this reaction because you're trying to capture that valuable ammonia to get the nitrogen, right? Trap the valuable nitrogen that's in the ammonia, or you do this because you've got too much ammonia floating, you've got ammonia floating around your blood, and that's toxic. Okay, too much ammonia in your blood, what is it? It's BUN, which is cute. What does BUN stand for?
Oh,
I forgot what it stands for, but I always think it's cute because it's called BUN. But it's, if you come up with it, great. So anyway, so we're making a glutamate here, right? Okay, our glutamate, and we'll combine that with an ATP for this reaction. Our enzyme, and so you're going to need to know this reaction, be able to draw the structures, name the enzyme. All right, the enzyme is glutamate synthetase.
One direction.
One reaction.
Or one direction.
One direction. I don't really care. We're not going to talk about regulation of this one. To make glutamine, Plus, I'm going to put a phosphate over here. Okay, plus a phosphate. What's the ATP? We're not going to go do the mechanism for that, but what's the ATP doing in that mechanism? It's making a leaving group, right? We're going to put a phosphate on so we can knock it off. And then that glutamine, all right, can combine, can react with alpha ketoglutarate. Where do we find alpha ketoglutarate?
Oh, citric acid cycle.
Citric acid cycle, right? Got plenty of that around. Plus NADPH, our buddy NADPH that's used to synthesize things, right? And there's another proton here. And that's going to make us 2-glutamate. And that, okay? So if we want to go through here for the names for these, right? I'm not going to name them. I'm just going to give us out one-letter codes. So we've got a glutamate, a glutamine. Our glutamine's over here. We have alpha ketoglutarate. Unfortunately, there's not a one-letter code for alpha ketoglutarate I could make you memorize.
Unfortunate for who?
For me. So this is our reaction. If we add these two reactions together, what our overall reaction is we have ammonia that comes in, right? I make a glutamine, but then the glutamine goes away, right? I make a glutamate. I start with a glutamate, but one of my glutamates go away, right? So it's gonna be the overall reaction is NH3 plus alpha ketoglutarate plus all our energy cofactors. So we're going to spend an NADPH and we're going to spend an ATP and we go to make a glutamate. And then we end up with an NADP plus and an ADP and a phosphate. Okay, so that's our overall reaction. And I need you to know this enzyme. This is glutamate synth. Oh, I didn't write it down. I think it's glutamine. Yep, I'm gonna have to give it to you next time. Okay. Glutamine something, because I didn't write it down.
Is it in the textbook?
It is in the textbook.
Synthetase?
Is it a synthetase?
Wouldn't you?
From my
It should be-
notes from last year, it's
it-
glutamine synthetase.
It can't be a synthetase because the synthetase means it uses ATP, so it'd be a synthase.
Glutamate synthetase.
Glutamine synthase.
It says glutamate.
Does this say glutamate?
It's glutamine up on the top, but also
Oh,
I'm sure.
this should be glutamine synthetase, correct? Because we're making glutamine.
Yeah.
And then it's is it glutamate synthase?
Yes.
glutamate synthetase.
It's synthase. That's what the textbook says.
And then glutamate synthetase.
Like glutamate.
Glutamate. So we're fortunate, even though I don't remember this, that we're fortunate that these reactions are actually named, if we got those right, with what they actually make as opposed to the reverse direction, which
That's what
sometimes
the
happens.
textbook says.
So glutamate synthase, I'm certain of that, the synthase, because this doesn't use ATP. And then glutamine synthetase because it does use ATP. That's what the synthetase versus synthase means. In lab, we are working with methynyl tetrahydrofolate synthetase because it uses ATP. So I've never seen an exception to that naming. Okay. Questions? You guys can leave. If you have questions, come on up and ask.
Where's the